Good morning, I'm Giovanni Stilo from the University of L'Aquila, and today I will present my work entitled Predicting Disease Genes for Complex Diseases using Random Watcher-Walker. First of all, let me introduce the problem that we are going to fight. Consider a PPI network, as the COVID-1 reported on the left side of the slide, where every node could be associated with one or more diseases. The disease gene prediction problem consists in to predict the disease related to genes in a situation of high incompleteness, where only the 10% of human genes have known to be associated with a disease. The main reason to predict new disease gene association is to speed up the research by driving new in vitro tests. To do that, we developed a network-based methodology to jointly learn functionality and connectivity patterns surrounding protein in the PPI network. Let's start by introducing the main contribution of this work. First of all, we propose a novel framework for disease gene prediction based on a variant of random walks named Random Watcher-Walker, RV Second. The conducted experiment showed that exploiting connectivity properties alone is not sufficient to reliably identify disease genes. A careful selection of disease boosts predictive methods to work with highly incomplete protein-protein interaction network. As it's possible to see in the current slide, the network model of the labeled PPI can be described as follows. Each model represents a protein, edge protein-protein interactions, where the nodes marked in gray are those that belong to one or more diseases. Associated to every node, we have also a feature vector that contains the associated disease, as it's possible to see in the top right part of the slide. Furthermore, the disease modules, the nodes associated with a certain disease, are highlighted with colors red, green, or blue. Let's take a closer look to the feature vector. Consider the vector related to the node V, since the vector and disease are indexed. If disease I is associated with protein V, then the corresponding cell I is valued with number 1, as it's possible to see in the slide. Let's take now a look at the overview of the proposed approach. The proposed approach first collects functional structural connectivity patterns, then we learn embeddings for disease labels, and at the end we train a neural network. The first phase allows to collect network connectivity patterns using the random watcher-walker. The walker, that is on one node V, pick at random one label from the feature vector of node V. Then the walker jumps with uniform probability to one of the neighbors of V. In that way, the performed random walks embody both functional features of the traversed nodes, disease labels, and structural features, connected proteins in the PPI network. The intuition behind that is that by collecting a large set of walks, frequent patterns will naturally emerge. In this example, we start our random watcher-walker visit from node M by select a random D2 between disease D1 and D2. Then we can jump to one of the neighbors of the node, such as H, L, E, and B. Suppose we chose node B. Then again, we need to randomly choose between disease D2 and D3. D3 was selected. Then we move forward, jumping from one node to another until we finish our visit. And the sequence of labels was produced as in the slides. D2, D3, D3, W, D2, D2, D1, D1. Note that not all the nodes have a disease. In that case, we are going to choose the wildcard label double. The second phase allows to learning emerging pattern as context of individual labels. To do so, we applied the deep learning model skipgram to encode these patterns into a dense representation. Skipgram is a popular NLP framework employed to learn word embeddings. In our case, sentences are the random watcher-walker sequences. In that way, it was possible to encode the latent relationship between node label, diseases, and the network topology into a dense node label representation. Subsequently, the label embeddings are used to enrich the feature vector of each node. Each corresponding cell, valued in the initial vector, is replaced with the computed embedding of the corresponding disease. Cells that are not valued are replaced by a zero vector of the size of the embeddings. For this reason, from now on, we consider a feature vector as a matrix. In the third and last phase, we design a neural network that takes in input the protein features matrix, the enriched feature vector, and returns the probability that the chosen protein is associated with a certain disease. The architecture of the neural network is composed of two fully connected layers, where the last layer uses the softmax activation function to compute the probability. We test our method against three baselines. The first one is the baseline method without embeddings, that only considers functional patterns. Diamond is the second baseline, was proposed by Barabasi et al., is commonly considered the state-of-the-art and most cited study on disease gene prediction. Diamond exploits only connectivity information. The third baseline is the random walks with the start, proposed by Köhler, that, like Diamond, use only connectivity information. To test our method, we use two relevant datasets in the network medicine domain. On the one hand, we employ the Diamond dataset to make a fair comparison with the state-of-the-art. On the other hand, we test our method on a very reliable PPI network with low bias, HI third. Whereas several disease databases can be employed to label the PPI network. We choose the disease categories employed in Diamond, DisGenNet, and those used by PhenoRank. We obtain four possible combinations of PPI networks and disease categories that expose different characteristics, as it is possible to see in the reported table. To evaluate our system, we split the evaluated dataset into two parts, 80% for the training set and 20% for the test set. We ran our experiments 10 times and we averaged the results together. Note that the common evaluation measures such as precision, accuracy, and F-score are ineffective in this domain, since there is no assessed experiment method to create negative example without introducing noise. Moreover, since the output is probability vector of disease categories, we don't get a single answer, but a ranking. Then we use the recall at k measure to test our system. Note that the recall at k is the probability that the right diseases is predicted within first k position of the ranking. As example, recall at 1 measured the percentage of times the right disease is predicted in the first position. In order to avoid trivial learning and correctly test our system, we need to process our dataset. As it's possible to see in line 2, 3, and 5, we have a simple situation where we have just one disease associated with the protein. In that case, it is enough to remove the disease from the input. A more complex situation consists of an instance with associated more than one disease. In this case, we need to remove alternatively one of the diseases and keep the others, as it's possible to see in example 4 that became 4A and 4B. This table shows the result of each combination of PPI datasets and disease dataset. It is possible to notice that, contrary to Diamond and random walk with the start, Our method exploits both node attributes and connectivity features, which systematically result in better performance, using only similarity of the feature vector, as is in the baseline methods, that not allow to learn regularities. In other terms, comorbidity alone is a weak predictor of disease genes. And also demonstrate by looking at the first column and by Agrawal et al. That random walk with restart does not perform worse than diamond, on the contrary. To conclude, the random walk watcher walker has the ability to discover specific pattern of connectivity and functional features that are in the proximity of the nodes related to a given disease. The conducted experiments highlight the existent correlation between the achieved performance and the used PPI disease categories. In agreement with Agrawal et al., we have shown that connectivity features alone do not allow to discover disease modules in an effective way. That's all. Thank you very much for your attention, and please visit our lab website for further information. Thank you.